editing allows us to create new and more effective types of pest control with less impact on the environment. Many people have questions about how gene editing works. In the Scott Lab at NC State University, we often use a technology called CRISPR to edit genes. CRISPR has two important components. One is a protein, and one is a short piece of RNA. The protein is like a pair of scissors. It cuts long strands of DNA into two pieces. The short piece of RNA acts as a guide. It shows the CRISPR-associated protein where to cut. By using pieces of RNA with different sequences, we can guide the CRISPR-associated protein to cut precisely at a specific place in an organism's genome. Now, if a piece of DNA is cut in two by CRISPR, that presents a problem for the cell. DNA that's all cut up can't work right, so organisms have methods to repair cut DNA. One way a cell repairs cut DNA is called non-homologous end joining. Proteins from the cell take each cut end of DNA and stick them back together again quickly. This solves the problem for the cell, but it can lead to small deletions or insertions at the cut site. You may know already that a strand of DNA is made of many subunits, and each subunit is one letter, A, G, C, or T. Non-homologous end joining can sometimes drop letters that were in the DNA or stick new ones in. A small change to the sequence of letters can have big impacts on a gene's final product, or make it stop working entirely. Edits like these can be easy to see in our fruit flies. At the Scott Lab, we use some lines where gene editing has given the flies different eye colors. Another method cells use to repair cut DNA is called homology-directed repair. Homology-directed repair is when the cell uses a template to recreate the sequence of letters near the cut site. Our flies have 2 copies of every chromosome except sex chromosomes, so there is usually a perfect template to copy. With this method, an organism can avoid short insertions or deletions. But we can take advantage of this pathway by providing our own template to copy. We can send a DNA template of our own design with the CRISPR components so that homology-directed repair at the cut site uses our sequence instead of another copy. And if our sequence includes new genes, the cell will incorporate those genes into the repaired chromosome. Now we can edit genes. We can change how genes work or add new ones to our flies in the lab. But how can this help us with pest control? We need a clever plan to make use of gene editing technology. We can take advantage of the way genes work to create new genes that make the components of CRISPR on their own. If we insert genes for CRISPR RNA and CRISPR-associated protein into a fly's chromosome, then its offspring will have genes that allow them to make edits at the genes that we target using the same CRISPR cutting activity. Imagine we inserted genes for CRISPR RNA and CRISPR-associated protein into a precise spot on chromosome 5. Now imagine that the CRISPR RNA which we inserted guides more cutting to the exact same spot in subsequent generations. If a cell uses homology-directed repair, then the only template the cell can copy to fix the cut is the sequence including the genes for CRISPR RNA and CRISPR-associated protein. A fly that started with just 1 copy of those CRISPR genes on chromosome 5 now has 2, and that fly will pass those genes to all of its offspring. This can cause a gene to spread quickly through a whole population. The system is called homing gene drive. With this system, we can get new genes into wild populations. These genes can help us keep pests out of our spaces by affecting fertility or causing some flies to die off. Or, for disease vectors such as Anopheles mosquitoes that transmit malaria, gene drive can be used to spread genes into the population that prevent the mosquitoes from transmitting malaria to humans.